Dr. Vett Lloyd, Associate Professor in the Department of Biology at Mount Allison University.
So what we do is we work on epigenetics. So epigenetics is a topic that may sound fairly arcane and erudite to a lot of people. So to give you some concrete examples of what we do is we're working with 4 major model systems. Drosophila, everyone's favorite little lab buddy. We work with a fungus that attacks wheat, so that's very important in terms of making sure people still have food to eat. We work with ticks. Ticks are very important in terms of being a vector for pathogens. Lyme disease is the best known of those pathogens. And then we work with cancer cells, which don't really need much explanation in terms of human health. We've had progress in all of those fields. The cancer work, the fungus work, what we're doing is we're trying to modulate epigenetic gene expression. Epigenetic modulation allows us to control the behavior of the cancer cells. Probably the major advances have been in terms of the Lyme disease research. We've made a lot of progress this year finding new sites of risk within New Brunswick. So to use the Lyme disease as an example, one thing we've been doing this summer is I've had a field crew, the tick patrol, go out through the province, dragging little fuzzy blankets across the province, which sounds silly. What we're doing is we're finding ticks, so we're monitoring where those ticks are, which translates directly into a risk to human health. Now, based on that, we've made a very detailed map of where the high-risk areas are so that the message can now go out from public health to be extra vigilant. Another major accomplishment would be our analysis of exactly what Lyme disease bacteria is present in New Brunswick. The tests that are designed to figure out if someone has Lyme disease or not, there's a simple blood test. It's designed to pick up an American strain of the bacteria because that's where the test originated. The test is testing for the wrong strain of bacteria. And this is a very simple graph here. These white strain is what the test detects. The black strain is another strain that the test doesn't detect. Mostly in New Brunswick, we have either the black strain or called N40 or a hybrid of the two. New Brunswick Health Research Foundation has been very generous in terms of supporting students working in my lab over the course of not just this past year, but previous years. That's an invaluable experience because frequently when you have federal grants, yeah, it's wonderful, but by the time you've bought all the reagents, you can't pay summer students. So that allows students to get into the lab, to actually experience research. They can go on then to do their senior undergrad year and honors degree. Many go on to do graduate work. So it gets them into the lab early so they can see what this research stuff is about.